So, okay, we're going over the unit exam, unit 1 exam. I'm not going to post this until after the last person is taken their exam. There's still one person has to make up the exam, so I forget.
Kylie.
Oh, yeah, it was Kylie Osario. So anyways, scientists examined the folded structure and purified protein and resuspended it in water and found that amino acids with nonpolar groups were primarily buried in the middle of the protein, whereas amino acids in the polar groups were primarily on the surface of the water of the protein. Which of the following best explains the location of the amino acids in the folded protein? Well, first of all, let's remember what a protein is. It's a polymer of amino acids, and remember that that means that you have these NH2, central carbon, hydrogen. Then you have this R group that sits out here, and it's a variable, so there's various ones. And then N amino acid would have a double bond OH or carboxyl group. But then when you link them, when you take two of these and you link them together, they form a peptide bond. And that peptide bond means water leaves, an OH leaves, and an H leaves in the form of H2O. Then you have what's left over is a carbon bonded to the nitrogen, a double bond to this oxygen, and a hydrogen sticking up here. All right, so you have to count the bonds, but you'll see that there's 3 bonds in the nitrogen and there's 4 bonds in the carbon, so you're verified that you're correct. So anyways, there'd be a bunch of these all linked together. Let me, instead of doing, and they would be in a long chain, correct?
Mm-hmm.
All right. This would be R1 and this is R2. So I'm going to simplify the drawing. Instead of adding all the atoms, we can say, well, I'm going to put the proteins as blocks, if you don't mind, in white, representing the white that you see above. And then they're bonded through a peptide bond to each other. And then sticking down, you have an R group with different properties. And some of the R groups, as you were asked to study, different R groups have different properties. So R group 1 might be nonpolar, R group 2 might be polar. That's the easiest of, when you're talking about a protein folding, that's probably the easiest to visualize. So you got all these R groups sticking out. Now, if you take this chain, and it's really long, I'm just drawing a few of them, and you put it in water, and I'm going to change the color of these. I'm going to change 2 of them to blue, and I'm going to leave 3 green. Okay? Let me erase. Where's the eraser? There it is. So I'm going to change this one and this one to blue. Let's change 3 of them to blue, just because, and 2 of them will be in water. So what'll happen, what do you think is going to happen when you take these amino acids in a chain and put it in water when these are hydrophilic and these 2 are hydrophobic?
The hydrophobic ends up in the fold.
They're going to fold inward, right?
Yeah.
And so what you'll end up with is some kind of conformation where you have an outer ring of these R group, the blue R group sticking out, and an inner ring where the two hydrophilic or hydrophobic are inside. And you'll have something like this going on, right? Where these are the blue R groups, and those 2 are the green R groups. Does that make sense? They wouldn't necessarily be a closed circle, but They look something like that. That is folding, and it's because of hydrophil. Hold on a second. So what would happen is that what you would expect, and what does happen is that the hydrophobic portions would bend on the inside, the hydrophilic on the outside, and that's because of intermolecular forces. I'm going to write down the words that they love to use: intermolecular forces, the forces between molecules, and intramolecular forces. Forces inside a molecule. Well, this is obviously one molecule now, but it's hydrogen bonding, or not hydrogen bonding, I'm sorry, but hydrophilic / hydrophobic interactions. It would be. Does that make sense to you guys? Mm-hmm. All right. So let's look at the answers. 1, R groups on the surface of the protein can form ionic bonds with the charged ends of the water molecule. Well, 1, water is not charged. Water is polar. So this is partially charged. Partially charged. These are partial positive. That is not charged. Charge would be something like sodium +1 and chloride -1. Those are full charges. These are partial charges. These are polar. Often we draw that like this. The positive being the cross. All right? Showing where the electrons are being pulled toward. All right. Polar R groups are too bulky to fit in the middle of the protein and are pushed toward the protein's surface. Is it about size?
No.
We don't know what size these R groups are. They could be small or big. What they are is they're probably either they're ionic or they're polar. Either way, it's that hydrophilic interaction that's going to make them point towards the water. Non-polar groups cannot form hydrogen bonds with water and are pushed towards the middle of the protein. Perhaps that sounds like an okay answer. Pushed. Well, let's see what the next one says. Non-polar R groups form from different parts of a protein, form covalent bonds with each other to maintain the protein structure. Covalent bonds? Folding is not covalent bonds. Folding of a protein is a tertiary, folding is what happens because of these weak interactions. Covalent is a strong interactions. These are weak interactions.
Folding is secondary or tertiary?
It's secondary and tertiary. So folding includes secondary and tertiary, as well as quaternary, really, since you need to fold the various proteins. Remember, quaternary means more than one polypeptide. Let me just say. So more than one polypeptide, all right? That's when you get into quaternary structure. The example that we talked about with quaternary structure would be hemoglobin, and that's one folded protein here, another identical folded protein made from the same gene associated with it there, another folded protein here, and an identical folded protein on the other side, and they form this 4 proteins to make 4 polypeptides to make a single protein called hemoglobin, which is made of 4 polypeptides, 2 different proteins, 2 copies, two copies of copy 1 of gene 1 product, and 2 copies of gene 2. So there's 2 genes on the DNA. One makes the 1 and the other makes the 2. We call them alpha and beta. But in any case, this is when you have more than one polypeptide making a protein. We call that protein hemoglobin. That more than one polypeptide making a protein is interacting, making a shape. It's called quaternary structure. Tertiary structure is the folding of each individual protein. Secondary structure is the folding of the region of the protein. All right. So there you go. That's what we're talking about when it comes to this answer. So the best answer is C, right? Was that the answer?
Yeah.
All right. All right, so can we go to the next one here by clicking here, I think? All right, this next one, you got it right. It's glucose, galactose, fructose. It asks which of the following statements about glucose, galactose, and fructose are most likely. Carbohydrates have different properties because they have different arrangements of carbon, hydrogen, oxygen atoms. That's correct. The reasons the others are wrong, I think, is important to look at. It's the same properties. They have the same number of carbon. They do have the same number of carbon, hydrogen, and oxygen. That actually caught a bunch of people because if you count them, they all have C6H12O6. But because their arrangement's different, their properties are different. And the example I gave you in class is some people can't digest galactose, and so galactose is a sugar that's involved in the disaccharide lactose, and lactose is given to babies by mothers, and they can't digest it because of galactose. Glucose is digestible by anyone. If you can't digest glucose, you're likely never born. Fructose is difficult to digest for a lot of humans, so fructose should not be. Our livers have issues with fructose. So really, when we look at that answer, it is the best answer. And it was a trick question when they said, because the second half of that answer is correct, they're just looking to see if you understand that this arrangement means they have different chemistries. The reason we give each of these a different name is they have different chemistry. Fructose, galactose, and glucose do not act like each other in the body. All right. Does that make sense? Okay, so let's go on to the next one. Here we have grass with plant starch, and we have a picture of starch. It's a very simple picture with only 3 of them. You see there's the glycosidic bond, the CO, there's another C, the 6, you see the 6 6-sided ring. You have oxygen at the top, carbon 1, 2, 3, 4, 5, 6, right? This is glucose straight across. Then there's the rabbit. The rabbit eats that, and the rabbit makes this. What is this? Look at it and tell me what it is. So I'm going to draw that here in the, and I'm going to copy what we already drew.
You talked about a peptide, and I wrote that down.
And I'm going to go ahead and copy this thing. I wonder if this is going to record it in such a way as not to overlap this. But we'll find out. I'm going to copy it, then I'm going to delete it. In the notes, you have a C. You have a C. This is one of those things you had to know. These molecules, C double bond O, OH. So there's a carboxylic acid group here, an acid group here. Then I'm just going to draw a line drawing. I'm not going to put the C and the H's because I don't want to spend the time doing it. But the last one, I'll put the C and the H is a C, H, H, H. All the rest of these have single bonded carbon and hydrogen. What is that thing?
I can't remember what it's called.
So anytime you have, what molecule, the four molecules that you had to know, what molecule has mostly carbon and hydrogen? Mostly carbon and hydrogen. There's some oxygen, but mostly carbon and hydrogen. So let's go down the list. Could it be a protein? No, because a protein would have to have nitrogen, right? A protein has nitrogen, carbon, hydrogen. No protein has phosphorus, but sometimes sulfur, and of course oxygen, right? Proteins have that. We just drew it, right? Nucleic acids. Well, they do have the, there's the acid parts. You might be thinking acid because you have a carboxylic acid group. But remember, you have nitrogen, carbon, hydrogen, no sulfur, oxygen, and phosphorus. So that only has it's not mostly carbon and hydrogen. You built DNAs, and so you should know that this is not DNA. You know this is not a protein. So it only leaves you two options. A carbohydrate. So you're right that it's CHO.
But this wouldn't be a carbon.
It's not. Why is it not? Because it's not just CHO, it's what? At what ratio? 1 to 2 to 1. So it's not a carbohydrate because this is mostly carbon and hydrogen. Right? Look at it. You have 1, 2, 3, 4, 5, 6, 7, 8, 9. And you only have 2 oxygens. So you have something like 12 carbons or 13 carbons and 2 oxygens. And then, what is it? 26, 30 hydrogens, right? So it's mostly carbon and hydrogen. So it's not a 2 to 1. So let's go through the list. It's not a carbohydrate. It's not a nucleic acid. And it's not a protein. So it's the only option left. So, right, amino acids would be protein, yeah. So what's the other option? It sounds like there's some more work. Maybe index cards might help, right? So, because lipids, there's only 4 of them. It could only be one of 4. So you got to know these 4. It's a lipid. It's a lipid, specifically a type of lipid called fatty acid. So it sounds like it's a memorization thing. You got to know these 4 groups, right? You got to know not just the 4 groups, but what elements are in each one. So it's a fatty acid. It's a lipid. And why is it an acid? Because it has the acid parts here, and the fat part the lipid part is here. So this is fat. Fat is carbon and hydrogen, and acid is a carboxylic acid. Now, you should have recognized that shape because you should recognize this. I'll draw this up here. Phosphate.
I was thinking it was like the saturated, unsaturated.
The saturated, unsaturated fatty acids. Correct. You're absolutely right.
Oh.
So that's absolutely right. And this is a
Phospholipid.
Phospholipid.
So
Right?
is that
So this is what that means. Those little wiggly lines are that carbon-hydrogen zigzagging.
Is that
The hydrophobic, this is a hydrophobic tail. That's right, this is the hydrophobic one. And this is not a protein, so this is just hydrophobic tails. And this is the hydrophilic end, the phosphate.
Right.
The tail and the head. So this is not a protein, so there's no, it kind of folds, but we don't really talk about folding with lipids. Let's just say, or at least not usually, let's keep it separate, okay? So that's a phospholipid. All right, so lipids have those carbon hydrogens, and they're nonpolar. All right, so let's look and see what the question says. You got it right. It says, which of the following statements best describes how organisms such as rabbits obtain carbon necessary for building biological molecules? Rabbits eat plants and break down plant molecules to obtain carbon and other atoms, and they rearrange it into new carbon-containing molecules. Which you can see even if you didn't know that the starch is sugar and this is a lipid. But that would have helped you clarify the idea had you remembered that. because this is sugar or carbohydrates and you're making fat. That's what we do. We do that all day. All right, let's go on. Next one. I wish you just look at the ones you got wrong, so not worry about all these others. Cohesive property of water with 5 options. Okay, which of the following is responsible for cohesive property of water? Hydrogen bonds between oxygen atoms of one molecule and hydrogen atom of another molecule. Do you agree?
Mm-hmm.
That is correct. Slide. Come over here.
Water molecule, like you would do the big O, the 2 H's, and you connect the H's to another
Good,
O.
you got that. That's good.
And then you connect the O to some more H's.
Excellent. But let's remember that the reason it's called cohesive, and you're absolutely right, the hydrogen bonding is going on between the oxygen and the hydrogen, because oxygen is pulling the electrons more towards them in that direction. So this is partially negative, and because it's pulling, the oxygens are pulling, and this is partially negative, this one's partially positive, and opposites attract.
Opposites attract.
That's hydrogen bonding.
Wait, so can I see? Because this is how I drew it.
Well, hold on. I that's good that you do that, but I just want to point out this is called before I give it to you. This is cohesive.
And adhesive would mean like the water would go to other molecules.
Exactly. Co means together, to each other. Adhesive means stick, right? And so adhesive, ad means to other, and adhesive means stick. So it sticks to others. But see, it's the same thing. If you have a surface of something, let's just look at
Surface tension.
yeah. Well, surface tension is because of cohesive. They're sticking to each other. But if you have like
Yeah, that's the Onaconic atom.
yeah, if you have a plant, let's just use green because it's a plant. If you have a plant that's sticking up and you have these little things called, if you ever cut a plant open and has cell walls, it's not all, there's a bunch of little tiny capillaries, little tiny tubes that go right up the stem. If you could go just right up the stem, so this would continue up the stem. All these would be tubes, are made just these long tubes that go right there. The plant cell just makes these long tubes. So if you can use this as a straw. This is like nature's straw. Okay, these are all little tubes. So water sitting out here, OHs, being able to hydrogen bond, will start to stick to the side of the negatives will be attracted to the negative side of the capillary. The positives will stick to the positive side of the capillary. So these forces will kind of wiggling their way. Remember, these molecules are moving back and forth because of kinetic energy. And what happens is that hydrogen bonding pulls one water molecule after the other up the chain. So they just pull each other. This attraction between the hydrogen and the oxygen starts to pull one molecule after another right up the straw, up the stem, all the way out to the leaves at the top. Remember, we call that transpiration. All right, so this is let's say it's a bamboo plant. If you ever looked at bamboo, it's just a bunch of little tubes that's connected, that when you put it in water, you can grow bamboo right in water. It's connected, and the tube goes right to the leaf. And then water leaves, H2O leaves. Every time an H2O molecule leaves, it creates a vacuum that leaves space for water to move up the chain. So water constantly gets pulled up. That's why if you're going to water something, you want to put the water at the bottom and let the plant pull it up. Watering the leaves does no good because no water gets in. It'll come, if you want to get water into the plant, you want it in the roots, not in the leaf. The leaf is where water comes out. Okay, you want water going in through the roots. So that's an adhesive property. That's sticking to other things. In this case, it's sticking to the plant. Does Does that make sense?
Mm-hmm.
All right, let's go to the next one. So good. Did you get that? Did you want to do? You wanted to say something earlier about cohesive in your drawing. Did you want to try to draw it?
Yeah, this is how I drew it. I just don't know if it's right, but I drew it like this.
Don't drop baby. Drop baby. I have a heart attack.
Oh, what? Oh. Oh.
I can see where you're going with that, but let me show you just to point out a couple small errors, okay? It's hard to do this and do it accurately because what's going to happen is, I'm assuming you mean these are hydrogen bonds between them?
Mm-hmm.
It looks like they're bonds, and that's not happening because hydrogen only makes one bond. Does that make sense?
Yeah.
So it's just a small point, but it can be a big deal if you're not careful. It can give you some misinformation when you're trying to take a test. So what I would do, obviously you're not going to have a different colored pencil or pen or anything, but first I'd use a different color, and secondly I would just make it a wiggly line like that. So you're right, that's what's happening. That's absolutely right. And except that this molecule here would also not, that needs its second hydrogen, you just kind of drew one. And this molecule here, also you drew one hydrogen, right? It's two hydrogens. And because the hydrogens are both positive, they're kind of going to push away from each other.
Yeah, so they wouldn't share. They wouldn't share a hydrogen.
They do not share hydrogens, correct. And so what would happen is that you'd get the oxygen pointing this way, and they're going to be revolving, pushing each other around. So water molecules are constantly twisting and turning and spinning, and you'll have something like this. And in between these, obviously, this is three-dimensional, so in between this, you'll have another water out there somewhere doing that, pointing away from. So we would just kind of go like you would draw, if you want to point it something like that. So going away from. And then another one, point towards you, whatever. So there's, in 3 dimensions, you just want to make sure that you see that the hydrogen bonding is happening between oxygen and hydrogen. You see that? Mm-hmm. And they form this lattice, which is why when it freezes, it makes a snowflake, right? Because it has that geometric pattern. Because no two snowflakes look alike or whatever, which is I think somebody's proved that was wrong. But they make these geometric patterns because they're hydrogen bonding to each other in this kind of regular way. All right? Mm-hmm. So just be careful with the drawing there. That's the only thing I would caution you with. Now here it says, a typical bag of fertilizer contains high levels of nitrogen, phosphorus, potassium, but trace amounts of magnesium and calcium. Which of the best matches the fertilizer component with a molecule in which it will be incorporated in the area? So it's nitrogen, phosphorus, and potassium. Nitrogen, which of the molecules that we know of, we just did it a slide ago, has nitrogen, phosphorus, certainly. Nitrogen will be incorporated in nucleic acids. Well, that's true. Phosphorus will be incorporated in amino acids. That's not true.
I don't know why I clicked that, because I knew that nitrogen was right here, so I think I missed it.
Well, nitrogen is definitely in nucleic acids. Nitrogen is also in amino acids, but phosphorus is not in amino acids. Potassium is not in lipids.
Magnesium is not in carbohydrates.
That's correct. So because carbohydrates are mostly CHO and lipids are mostly CH, some O. There's some phosphate and some lipids as well. And nitrogen. Now, how could they change this question for your real exam? They could put phosphorus in lipids. That would also be correct. All right? So you got to be careful with that because of phospholipids. All right, next one. If 30% of the nucleotides in a single RNA strand are adenine, what percent are expected to be thymine in RNA? 0. That's right. You were right. Everybody that got this wrong made the same mistake. You read, in fact, I'll be honest, when I first read this, my instinct was to go with 30-30 because that's Chargaff's rule, right? The A and the T's equal each other, then G and the C's equal each other.
A and U are an RNA. There is no T.
That's right. There's no T in RNA, and they were testing. I really feel these kinds of questions are more reading questions than content, but it just goes to show you that you have to read carefully. I think that was just a mistake in taking it easy and reading. I know one of you needed to leave, not stay so late. Do you guys want to finish this next week? Another half an hour next week? I think we could finish the whole test.
I'm cool with staying.
Or you can stay now.
I'll just stay. I mean, I had to go to practice, but I already told my coach
Okay.
I'm not going to practice.
All right. I don't want to keep you from your life. Also, it looks like it's going to thunder and lightning out there, so be careful. Which of the following correctly illustrates a dipeptide and an amino acid in the optimal position to form a tripeptide? So what's a dipeptide? Back 2, correct. I'm going to go. And I'm going to, it said it right there, yeah. And there it is. There's a dipeptide, right? And I said, why do I say it's a dipeptide? Because there's 2 peptides, right? There's 2 amino acids. 2 R groups is a good way of looking at it. That's correct. Because each amino acid is going to have its own R group. So 1 amino acid, 2 amino acids, right? So that's a dipeptide. If you're going to connect it to another one, what's going to be over here? What needs to be over here is another N. They need to line up this way, right? If you put a carboxyl group next to a carboxyl group, it won't work because you'll have OH. Let's go ahead and do it just to show you. If I orient it the other way and I put the carboxyl group over here, now if I try to do dehydration synthesis, I have 2 OHs. Well, I only need 1 O and 2 H's. I need H, not OH and OH. So that really doesn't work that well. So so that's one thing to think about. The other thing to think about is amino acid polypeptides are lined up from the end terminus, the N terminus, the N side, right? And then the carboxyl side, the C side, or terminus. I believe that's how the book discusses it, right? C terminus and N terminus. So you got 2 N's. So if you're going to connect them, N to C, N to C, N to C, then this is N. Here's an N. And there's an N. So what's the next one? An N. And then when you have it, you have your NH. If you go ahead and put it, and you're connected to central carbon, hydrogen, R group, and then your C double bond OH. Then you see that OH and H can leave, and there could be a bond between them. So you're going from a dipeptide, 2 amino acids, 2A, tripeptide, 3 amino acids. So this will be number 3 here. 1, 2, 3. All right, so then this one, R group, N, N. There's the 1 H, the 2 H's. There's 3 bonds this N, 3 bonds this N. This looks right. 2 R groups, carboxyl N, amine N, carboxyl N. The first A looks right to me. Here, if we look at here, this carboxyl to carboxyl, that won't work. We just looked at Y. Here's amine to amine, also won't work because, yeah, you have the hydrogens, but where's the oxygen coming from to make water? There's no oxygen there. And then this one, again, that's missing. This is not even an amino acid. So, yes, there's an R group there. It's missing the nitrogen. This C should be a nitrogen. So they tricked you on that one. So that one is absolutely not right because this isn't even an amino acid.
Are they allowed to be backwards too? Like, you see how that one starts with the H and N, but that one starts with the double bonded oxygen?
What do you mean? Here and here?
here and here, like you see how this starts with that, but that starts with that.
Yeah, you could start it. You could line them up that way. So that's a good point. I should draw that. You could start with C double bond O on this side, right? And OH. That would be, usually it's not drawn that way. Let me say that. But could it? Yeah, it could be drawn that way. But what would happen is that when I connect them on this side, I'd have to have what? If this is the C, what's on this side?
H.
Right, it would be the N, right? The N terminus. So the N terminus has to be on this side. The C is on this side. So then this would have. That's right. You were right. It would have to have. It's connected to an N. Well, actually, that's not true. My bad. I left out. That is true. So it's NHH. And then over here, though, what would have to happen is you'd have to have another C double bond OH. Does that make sense? Because if you have C on this side, if C's on this side, N has to be on this side. They have to be. There's a direction. Remember the whole conversation we had about direction and language? Direction. You have to have a direction in order for it to work. And so you end up with CCNHH. And then, of course, you get water coming out here. But we don't draw it this way usually. But, yeah, it can be back, quote-unquote, backwards, right? This is the way it's normally drawn. Most, like you see here, the correct answer is actually drawn this first way. Okay?
Mm-hmm.
Are we good with that?
Yeah.
Yeah.
All right, let's go to the next one. It says, amylase is an enzyme that converts carbohydrate polymers to monomers. You have amylase in your saliva glands. As soon as you eat a piece of bread, do you ever feel that tingly feeling in your mouth when you eat a piece of bread, especially when you're hungry?
Mm-hmm.
That's the enzymes in your mouth breaking down the carbohydrate polymer into a monomer. Glycogen synthase is one enzyme that involves converting carbohydrate monomers into polymers. So what I would do when I'm given a sentence like this, I would start and think, well, without even worrying about which carbohydrate polymer am I talking about? Is it cellulose? Is it glycogen? Is it starch? Well, first of all, draw out the idea that a polymer is a bunch of units connected. Just draw it simple. And then in the first example, you have some enzyme. And I believe the problem, I'm writing down what the problem is telling me, right? Which is one of the first steps to problem solving, correct?
Mm-hmm.
What are they telling me? They're telling me I have a polymer. And they're saying that I have an enzyme that turns them into monomers. So it breaks them loose. What are the monomers? Sugars, and we could talk about all that. And which sugar, it doesn't matter. Well, for this example, the enzyme's taking this polymer and making them into monomers. That's it. Now, you can get into details and call that hydrolysis, the breaking up of a polymer using water, because water is going in to each of these bonds. We took water out to link them together. We're putting water into each one of those to break them apart, because one's going to turn into an OH and the other one's going to turn into an H, and that's what's going to happen there. So in any case, that would be the process that we're drawing for the first one. The second enzyme does the opposite. So you're going to take these monomers and you're going to take these monomers and you're going to turn them into these monomers and turn them into polymers, right? So this is enzyme 2. Does that make sense? So you're going to take them and link them together. That process is dehydration synthesis. Which is the opposite of hydrolysis. So these get linked up by pulling water out. So put water in to break them up, take it out to put them together. So now let's look at the question. Which of the following best explains the reactions of these enzymes? Amylase aids in the removal of water. Oh, look, it's exactly what the problem was about. Interesting. So I didn't know that. So, no, but I'm very happy with myself. So hydrolysis is the adding of water, and then dehydration synthesis is the removal of water, putting it together by removing water or separating them. Lysis means to separate, breaking them apart by adding water. So amylase aids in removal of water molecule to break covalent bonds, where to break a covalent bond, we don't remove water, right? We add water. That's a covalent bond. So we add water to break it up. So that one's wrong. Is that clear why that's wrong? Amylase aids in the addition of water to break down covalent bonds. That's correct. In the hydrolysis, lysis to break up water to break up molecules. Amylase aids in the addition of water to form covalent bonds, whereas glycogen, well, the rest are wrong for various orientations of this problem. So you can mix up the answers to give you distractors in all different kinds of ways, but as long as you understand this basic idea that to break them apart, you're adding water, and to put them together, you're removing water. Then you write down what they're telling you, because they tell you glycogen is making the stuff, right? Glycogen synthase is putting things together, correct? So glycogen synthase is doing dehydration synthesis, where amylase is doing hydrolysis. Amylase is putting water in, glycogen synthase is taking water out. So there, you know that you can choose the right answer. In this case, the right answer was B. Are you good with that?
Yeah.
I'm very proud of myself that I guess that was what was it about. Well, this has a series of sugar phosphate, right? Sugar phosphate. You built this in like 3 different ways. And then you have a double ring nitrogen space here, a single ring nitrogen space here, right? You don't have to memorize it, but one is going to be A, the other T, one is G, the other C, if there were 2 of these, where A is double ring and T is single ring and G is double ring and C is single ring, I think. I always get those confused, so don't quote me on that. So represents a nucleic acid and a fragment that is made of 4 nucleotides put together. Let me ask quickly, can you identify if this is DNA or RNA? The clue here is you do not need to, I don't even know what the question is, but can you identify DNA or RNA?
Yeah, because DNA is 5 to 3.
No, they're both, DNA and RNA are going to be oriented 5 to 3.
Oh, man. Then I don't know.
So you're looking at DNA. DNA has a ribose that's missing an oxygen. So when I draw ribose, this is ribose, always the same, the one-armed guy. Here's each of these corners is a carbon. This is the prime carbon, second carbon, third carbon, fourth carbon, fifth carbon. So the 2' here is missing an oxygen at the second carbon. So it should have, and if it's missing an oxygen, it's deoxyribose.
So is this RNA?
This is RNA. How do I know? I look at this 1, 2, I look at the second carbon, it has an oxygen. The first carbon has a nitrogenous base, and that's either a single ring or a double ring, depending if it's AT or GC. So I know that's RNA. And the other clue that's RNA is it's single strand, because DNA is double-stranded, anti-parallel. So which of the following characteristics best shows a fragment is RNA? Oh, look, I did it again. RNA or DNA. 5 to 3 orientation of the nucleotide chain. No, that's going to be true of DNA and RNA. Identity of each nitrogenous base, that's true. If this one is A, if this one's T, and this one's A, then I know it's DNA, right? If it's U and A and G and C, then I know it's RNA. So definitely B is true, but maybe there's a better answer. Changes in the phosphate, on the phosphate groups? No, they're always going to be sugar phosphate. That backbone is a backbone. It's not going to change. The type of bond linking the nucleotides together, also not true. The nucleotides don't get linked together. The nucleotides are held together by hydrogen bonds, right? So they're never covalently bonded. So I guess hydrogen bonds, but that's true of any RNA interaction or DNA interaction. So the answer is B, the identity of the nitrogen spaces, and this is RNA. Are we good?
Mm-hmm.
All right. Is this DNA or RNA? Which one's DNA or RNA? Nucleic acid one is, first one's RNA, second one's DNA. How did you know?
RNA has a single strand, DNA has double strand.
That's right. RNA single. And by the way, RNA can come in any shape. They just make them the same shapes in this picture. Yeah, just to let you see that it's one versus two. RNA can form all kinds of shapes. Like proteins, they fold. RNA folds just like proteins. Well, so does DNA, but DNA makes a predictable shape. DNA and RNA are nucleic acids that can store biological information based on the sequence of nucleotide monomers. It shows, figure 1 shows a short segment of each of the two types of nucleic acids. Which of the following best describes the structural difference between DNA and RNA? Backbone of DNA contains deoxyribose, whereas the backbone of RNA contains ribose. That is correct, as we just discussed that. And they're going to test it over and over again. They could have asked single strand versus double strand. So you didn't even have to know what we talked about to answer this question. But yeah. The rest of these, DNA contains types of nitrogen spaces where RNA only contains two types. No. The rest of these are going to be wrong. I'm not even going to bother looking at them. What are these? Quick. Quick.
Lipids, phospholipids.
Well, they're lipids. Lipids, correct. There's some saturated and some unsaturated. This is what?
Double bond.
So it's unsaturated.
Unsaturated has multiple double bonds.
Oh, one or more.
One or more, and then saturated has
all single bonds. Now, among the carbons, you'll always notice that there's always a double bond at the carboxyl group because it's a carboxyl acid.
So then you have this. So wouldn't you know that it's unsaturated or saturated by the way that it curves like at the top? Right
Yeah,
there?
they're excellent. Yes, it's going to bend because the double bonds are shorter. That's correct. So based on the molecular structure shown in the figure, which molecule is likely to be solid at room temperature? Remember, we used butter as an example versus olive oil. Olive oil is unsaturated, so it bends, so it's hard to lay down, for them to lay down on top of one another. So they're always moving around, so it doesn't solidify easily at room temperature. At room temperature, butter lays down, the fats lay down one on top of the other, so they form solids much more easy. So it is palmonic acid is going to more likely to be solid at room temperature. Because the absence of carbon-carbon double bonds allows the molecules to pack together tightly. I think that's the right answer. Oh yeah, there's a check. It's the right answer. Okay? All right, let's look at the next one. I'm glad that the test was like I thought. Water and ammonia interact to form hydrogen bonds as represented in the figure. So there again, there's hydrogen bonding, right? They're pointing, they label it hydrogen. Hydrogen bonds happen between nitrogen and hydrogen or oxygen and hydrogen. It can happen between fluorine and hydrogen too, but in biomolecules, it's nitrogen and hydrogen or oxygen and hydrogen. Is that clear? Mm-hmm. Okay. So which statement best helps explain the formation of hydrogen bonds represented in the figure? That's because nitrogen is more electronegative, and so it's partially negative, and oxygen is more electronegative, so it's hydrogen's partially positive, and that attracts one other. So nitrogen is a partial negative charge. Hydrogen is attracted to the oxygen, and the hydrogen attached to the oxygen has a partial positive charge. That would be the correct answer.
That tripped me out because I need to remember hydrogen doesn't have a partial negative. Oxygen.
Yeah, And the way you got to remember that is that opposites attract. Opposites attract. And so, and also those dots are electrons. You see the dots?
Mm-hmm.
So if there's extra electrons, is that going to be more positive or negative?
If it's extra electron, it's going to be negative.
And that's negative. So you know that those oxygen and nitrogen have extra electrons. You see them there. That means that they're partially negative. These extra electrons make them partially negative. Where the hydrogen doesn't have any extra electrons, they're partially positive. Does that help?
Mm-hmm.
So make sure you think about that when you're looking at these molecules. And you can see that they draw them like that.
They drew it, so it was obvious.
Yeah, it's just that those electrons, you have to remember what they mean. Those dots are electrons. All right, let's go on to the next. I think we should just do one more. What do you think? And then I could record whatever's left. All right?
Okay.
And just pass it on to you.
I get a B for this, right?
You get a B after you finish the item analysis, which means you'll listen to the video that I sent. What number are we on?
13.
We're on 13 and there's 25. So you'll have to finish all the rest of the 25, whichever ones you got wrong, and submit a piece of paper or online. Explain which one you got wrong and why, and I'll explain each of the rest of the tests on a video,
Okay.
rather than keeping you here any later. All right, so let's look at this last one. There's obviously, this is what?
RNA.
And this is?
DNA.
And here you would expect what two nitrogenous bases?
A and T.
A and T. And what's holding these two together? These two strands?
Wait, don't say it. Hold on.
But
Oh, wait, I don't know.
hydrogen bonds.
Hydrogen, I knew it. Why didn't you say it?
99% of the time, the answer is hydrogen bonds. It's seriously, it's crazy. What pulls water up the plant? Hydrogen bonds. What holds two molecules together? Hydrogen bonds. What causes surface tension? Hydrogen bonds. Why does water melt? At higher temperatures than other molecules the same size? Hydrogen bonds. I mean, it's crazy. So hydrogen bonds the answer. So there's your AD, A, U, T, C, A, and G. Notice G's double ring, A is double ring, just what I said. I think I said that correctly. C is single ring, T is single ring, and U is single ring. Because T takes over for U, the only difference between T and U is you'll see that there's an extra carbon in T, which makes it not fit in RNA, but that's another story. Which of the following conclusions is most clearly supported by the representation of nucleic acid number 1 and nucleic acid number 2? Nucleic acid number 1 contains only purines, whereas nucleic acid number 2 contains pyrimidines. Purines and pyrimidines are either double or single ring. Let's be clear, it's always a double ring hydrogen bonding to a single ring. Whether it's single or double-stranded nucleic acid strands, if they're going to contain both double ring and single ring. So that's not the answer. Nucleic acid number 1 contains sugar ribose. Is that true? No, it's deoxyribose. It's DNA. And 2 contains ribose. Nucleic acid number 1 contains positively charged phosphate groups. No, phosphate's always negative. The group phosphate is a P, my God, with 4 oxygens. With every oxygen having all these what? What are these dots? Electrons. It's a negative, always negative. All right, so nucleic acid number 1 contains AT base pairs and 2 does not. Correct. In fact, that was asked twice on this test. Correct?
Yeah.
All right, good job. I have to finish with number from
13.
13 definitely.
How do you know it's 13? Where do you see the number?
She said it.
I know, but where do you see the number? I don't see it.
On my computer it shows, but it's like up there somewhere.
So it's the same order. Oh, there it is, yeah. So I'm on question 13. All right. And I have to finish all 25. More homework for me. Very good. 13, finish to 25.